Hello, my name is Dr. Roderick L. Rowe, and today I will be talking to you about other organelles. The first other organelle we will talk about is the mitochondria. The mitochondria is the site of cellular respiration, and it is in nearly all eukaryotic cells. The structure of the mitochondria is it has two membranes. It has an outer membrane and an inner membrane. The inner membrane will form these inner folds. The inner fold will be called a cristae. Do not get this confused with cisternae. So the cristae is going to increase surface area. The inner membrane is going to create two compartments. The inner membrane space that is between the inner membrane and the outer membrane, and the mitochondrial matrix, which is anterior to the inner membrane. Like I stated, the cristae is going to generate extra surface area. Why is this important? Enzymes are going to be embedded in the inner membrane. So therefore, by creating extra surface area, you can put more of these enzymes there. Therefore, you can generate more ATP. This is a transmission electron microscopic view of the mitochondria. This helped scientists draw the image to the left. The next organelle we will discuss is peroxisome. The peroxisomes are specialized metabolic compartments bound by a single membrane. It is going to contain a couple of enzymes, catalase and oxidases. The oxidases are going to remove hydrogen from substances and transfer them to oxygen. Therefore, it will make H2O2. This is called hydrogen peroxide. This is also poisonous. Once the peroxisome has made this poisonous compound called hydrogen peroxide, another enzyme called Catalase will come in and convert H2O2 into water and oxygen. Therefore, the peroxisome is going to help detox. So to me, it's like the liver. It's going to help detoxify these harmful substances that are in your cell. So when you take microbiology, there's a simple test you can do to see what bacteria produce catalase. Catalase is an enzyme that will break down hydrogen peroxide. And if catalase is present, you're going to see this bubbling sensation, just like when you pour hydrogen peroxide on an open wound. The open wound is releasing catalase. Catalase is inside of the cell. So when you cut yourself, a laceration, and you pour hydrogen peroxide on it, you're going to immediately break down hydrogen peroxide into water and oxygen. So therefore, hydrogen peroxide is not a good chemical to use on lacerations. You should use something like betadine or iodine or alcohol, something else that's not going to be immediately broken down into water and oxygen. Now we're going to look at plant cells. So the next thing we'll talk about is specifically seen in plants. It will not be found in animal cells. That organelle is called the chloroplast. The chloroplast contains the green pigment chlorophyll, and it also contains enzymes and other molecules that function in photosynthesis. The chloroplast is found in plants, but it can also be found in algae. The structure of the chloroplast consists of stacks of membranous sacs. This membranous sac is called a granum. Multiple stacks is a grana. Each sac that makes up the membranous stack is called a thylakoid. And also, all of these stacks are floating in a gelatinous material called the stroma. So the internal fluid interior to the inner membrane of the chloroplast is the stroma. So the chloroplast is one of a group of organelles called plastids. It is also double layered, just like the mitochondria and just like the nucleus. This is a transmission electron microscopic view of the chloroplast. This helped them to create the image to the left. This is another transmission electron microscopic view of that chloroplast. The image to the right is showing you that chloroplast also exists in algae. So it's not just found in plants. It is also found in plant-like protist called algae.